Welcome viewers. I am Professor S. Jeevan Chandra, Department of Microbiology, Chaitanya deemed to be University. I would like to explain in detail about enzyme linked receptors found in cell that are used for cell signaling. In the previous class, we have completed the G protein coupled receptor. Today, we are going to discuss about enzyme linked receptors. So, some receptor proteins they require intracellular enzymatic activity either intrinsically or extrinsically. That means they may be activating the nearby enzymes or their cytoplasmic domain itself having a enzymatic activity. Most of the transmembrane proteins they have this enzymatic linked activity. When these receptors interacting with their corresponding ligand, cytoplasmic domain is activated, which leads to enzyme activity or interaction with the enzyme, then leads to enzyme activity. But the ligand is a soluble signal protein. Actually, this gene regulation is very slow. That means it should be strong enough to reside on the receptor. What a type of receptors we have? Tyrosine kinase receptor, RTK receptor, tyrosine kinase receptors. RTKs are expressed on all types of cells, especially eukaryotic in nature. They found in all cells. They receive extracellular signals. The ligand molecules will bind together to the receptor, then leads to dimerization of the receptor tyrosine kinase. Single receptor, single ligand is not enough. Dimerization of both ligands, receptors will leads to activation. So generally there is a question arises. Really these receptors are not nearer together. They are far away on the cell surface. But there is a high degree of attraction towards these receptors. When ligand is attached to receptor, it acquires the affinity character. Now it will attracts other receptor which is interacted with the ligand, then both are dimerized together. Such type of activity is called receptor affinity. Upon dimerization, activation of kinase will be takes place in the cytoplasmic domain. So the interior side of the receptor is acquiring a kinase activity. What is a kinase? Kinase is an enzyme which acts on ATP and transfers a phosphate group to particular protein. Now, binding of intracellular molecule leads to intracellular signal protein accumulation. Now, protein phosphorylation will leads to activation of the intracellular signal molecules called secondary messengers. Now, secondary messengers will be carry out the biological function. After completion of the work, the protein will be having its phosphate should be removed. The protein phosphates removed by means of phosphatase group of enzymes. So kinases are the turn on group, phosphatases are the turn off group as like a switch. A protein having its phosphate is turn on, the done by kinase, removal of phosphate done by phosphatase enzyme. Intracellular proteins like phospholipase C, inositol triphosphate pathway, and other mechanisms like IP3 and RAS protein, CMYCG protein activation requires a RAS protein. These all are requires the receptor tyrosine receptor. What they are going to do? Inositol triphosphate will control the calcium channel as like G protein will do, and RAS protein will control the cell division, cell cycle G1, G2, G3, G0 like that. Cell cycle is controlled by RAS protein. Cell division, cell division. If RAS protein is getting mutated, the cell undergoes tumorogenesis. Cancer will be results, and thus it plays a major role in cancer. So let us cite how all RTKs will be activated. Most of the RTKs getting finally activates the RAS protein. A family of monomeric GTP binding protein is a trimeric of not trimeric like G protein. It is resembles certain activities like G protein, but will activating itself by GTP binding, inactivated by hydrolysis of GTP removal by phosphatase. But what happens? This RAS protein finally activating the MAP3 kinase, 3-tier kinase, MAP, mitogen activated protein kinases, mitogen, mitotic cell division generator, activation, protein, MAP, kinase, 3-tier kinase acting on the second kinase acting on the third kinase, like that they are going to be activated. The inactivating RAS protein can be activated by effect on inactivated one. Now RAS protein getting activated, cell ignores signal to divide. Now effect of permanent activation of RAS, cell act as constantly receiving the signal as a mitogen. So there is a key factor between these two RAS proteins. If RAS protein is inactivating, the cell ignores, there is no signal to divide. Cell do not divides. If if permanent activation of RAS is there, cell acts constantly receiving the signal, and as like a mitogen, it going to be dividing. So majority of the oncogenic proto-oncogenes, example CMYC genes, these all are presenting the unnecessary mitogens to cell. Now cell control over the cell system, and it will going to uncontrolled cell division and converted into tumor cell. That is in case of BCL2 receptor or either any CMYC genes. These all are going to be taking place. Mutant RAS will be having 30% incidence in all human cancers. So that leads to turn on of RAS protein, which constantly activating the mitogen activity. Now cancer is prevailed. So 30% means in 10 members 3 persons are having this type of profile. So don't need not worry about that one. But this is the common dynamics of the RAS protein. How it is going to be activated? See here, the signal molecule is searching for its homologous domains of RTKs. They receive the dimer signals, and now signal dimerization will take place. Cytoplasmic cytosol activating by phosphorylation of domains, autophosphorylation at tyrosine kinase system. The phosphate group is yellow colored phosphate group added to tyrosine amino acid on the cytoplasmic domain by autophosphorylation and cross phosphorylation. One domain should be phosphorylated, then it phosphorylates the other next domain by cross matching. Sometimes it is referred to be domain kissing. So one domain kisses the other domain kisses the next domain. Like that, it is a domain kissing. Domain kissing leads to transphosphorylation, activated receptor tyrosine kinase having a blinking. Now the domain kissing is leaded to activation of associated GDP protein. Inactive RAS protein is along with GDP. Now uh, it should be activated. So it acquires adapter proteins, RAF protein and RAS activating protein, RAS adaptation protein with SOS system. Now it is going to acquire the kinase activity. Now it removes the GDP with GTP. Now the enzyme kinase will be there. Now it going to activate the RAS protein. The activated RAS protein having GTP, now it translates the signal downward. So transmission of signal will be takes place. Now it will be diffusing the signal molecules downward. Activation of RAS protein requires MAP3 kinase systems. MAP kinase is first getting activated. Activated MAP kinase acting on the 2K MAP kinase. Previously it is 3 kinase. One kinase was removed. Now it phosphorylates the domain. Now second phosphate group is added to MAP. Now one kinase is removed. Then MA kinase is there. One kinase. Now it is going to diffuse it into different system, and phosphorylated MAP kinase will having signal divergence and control the protein X, protein Y, gene regulator protein and gene modulatory protein A, B. Now it will take over the two major events. Cytoplasm changes in protein activity. So some proteins present in the cytoplasm will be changed their conformation, either activated, deactivated. Activated proteins may be acting as a gene regulator protein A or gene regulator protein B. They diffuse it into nucleus and they're going to change the gene expression pattern. So they can control the gene expression. So extracellular space, previously the receptors were far away. When ligand is bind together, they come together. And how the RAS protein is getting activated without ligand, that means cytoplasm domain, the constitutively RAS protein mutant will unable to hydrolyze the GDP, so it continues the signals and multiplication in absence of signal leads to tumor genesis. అంటే ఏమవుతుంది, అవసరం లేకున్నా కానీ రాస్ ప్రోటీన్ యాక్టివ్ అవుతుంది. ఎందుకు అయింది అని అంటే majority of the RAS protein they contains mutations, the 30% individuals having this activity, so the tumors are generating autonomously. So some activated these systems are track into the nucleus and controls the cytokines and hormonal gene expression, and also it also controls the JAK-STAT pathway. This JAK-STAT pathway plays a major role in cellular survivability. And receptor kinase activation leads to receptor activated translocation of the nucleus proteins. These translocation to nucleus and they control the gene regulatory proteins which required for small pathway. And RTKs, receptor tyrosine kinases, also leading to tumor growth factor B superfamily, are very important for animal development from embryogenic stage to animal cell, because in zygote we are a single cell, but after embryonic stages we are transformed into an animal. So they require differentiation of different tissues into different organs and different organizations, so that's why their growth factors are required. Autophosphorylation of RTK then recruits activation of SMAD protein. The SMAD protein activates and releases a binding of different SMAD proteins to particular systems. They will diffuse it into nucleus and takes the regulation of the gene expression and gene activity in control over by these systems. So this is like this. When what happens, the receptor domains by JAK-STAT pathway. Now JAK tyrosine kinase is inactivated previously. When cytokines are binding to receptor tyrosine kinase exterior surface will leads to dimerization. Dimerization of cytokines leads to cross domain kissing. ఇక్కడ చూసారా, now one domain transphosphorating another, it is called domain kissing. Domain kissing leads to autophosphorylation and 4 times phosphorylate groups are there. Now activated CTD domain, cytosolic translated domain, will recruiting the start gene protein complex. Start gene protein complex, now it is activated together and diffused into dimerization of start gene will leads to activation. Now it is diffused into nucleus by dimerization and migration. Now they are going to searching for DNA of a particular location, and now along with the regulator protein, now it is going to activating the targeted gene. Now RNAs are transcribed. This is the how the tyrosine receptor kinases will control over the system. I 3 mechanisms are, first one is G protein coupled receptor linked to G protein, then cyclic AMP or IP3 inositol triphosphate diacylglycerol for calmodulin calcium receptor, and then regulatory elements diffuse into nucleus. One phase. Second phase, receptor tyrosine kinase activation leads to MAP3 kinase, MAP2 kinase, MAP kinase, then they leads to RAS protein activation. That is second step. This is third step, JAK protein. JAK-STAT pathway is controlled over by cytokines. Cytokines are the enzymes and protein molecules which are required for cell differentiation of T cell into B cell, B cell into antibody producing plasma cells or any other immunological cells, immunological recognition. So cytokines will activate the receptor tyrosine kinase, and domain kissing autophosphorylation takes place and they activate the phosphorylation. Now they recruit the start protein are getting phosphorylated. Dimerization of the start protein will leads to diffusion into nucleus, within the nucleus along with the regulatory element which is going to activating the genes which are required for gene translation. Now cell will be differentiating or controlling. Tumor growth factor beta, it is also requires tumor genesis by SMAD pathway. So activated tumor growth factor, again domain phosphorylation leads to SMAD protein. A SMAD protein getting phosphorylated, which is previously inactivated SMAD going to interacting with the uh, activated SMAD dissociate from the receptor, bind to SMAD and migrate to its own location on the nucleus. Now within the nucleus, SMAD going to regulate the transcription of tumor growth factors. So TGF beta is very versatile to promote the tumor growth. Once again, tumor growth factor beta interact with the receptor getting phosphorylated autophosphorylate activated receptor phosphorylate itself and recruit the activated SMAD protein. Activated SMAD protein which is dissociated from the receptor bind to a different SMAD protein associate and translocated into nucleus. Within the nucleus, particular chromosome will be open up by means of gene regulatory proteins. Now they open up the gene and undergoes transcription followed by translation. So now here we can envisage two important configurations. So the gene activity, gene regulatory proteins or targeting proteins of many cytoplasmic side are controlled over by two diversity mechanisms. What are they? Either G protein coupled receptor, either tyrosine kinase receptor. Activated G protein will diversifying the signal into two pathway, either cyclic AMP pathway, inositol triphosphate pathway. Adenyl cyclase also control phosphokinase A. Phosphokinase A also have target protein regulation and gene regulation activity. G protein going to activate the phospholipase C. Phospholipase C will activate inositol triphosphate diacylglycerol. Inositol triphosphate diacylglycerol has a two-way mechanism. Now signals are converged on phosphokinase C. Diacylglycerol activate the PKC, calcium activate the PKC. This is called signal convergence. Calcium directly promotes the calmodulin receptor. Calmodulin kinase also have gene regulation activity, target protein activation, signal divergence. Convergence can be carried by phospholipase C, inositol triphosphate, diacylglycerol targeting same protein called PKC. This is called signal convergence. But what about the interaction of lipase C? Phospholipase C can be directly or indirectly activated by G protein coupled receptor as well as the tyrosine kinase receptor. This is called crosstalk, signal crosstalk. Half of the domain sharing with G protein, half of the signals shared by tyrosine kinase. This type of activity called signal crosstalk. Signal crosstalk promoted by two receptor, G protein coupled receptor as well as the tyrosine kinase receptor. Third group, adapter proteins of tyrosine kinase activating the RAS activation protein. RAS activated protein will activating the MAP3 kinase, MAP3 kinase to MAP 2-tier kinase, MAP 2-tier kinase to MAP kinase. MAP kinase also have gene regulatory protein control and cellular target protein control. So 4 protein, phosphokinase A, calcium calmodulin kinase, phosphokinase C, mitogen activated protein kinases. These all are having two ways of mechanism, signal divergence to cellular targeting and nuclear targeting. So this is called how signals are going to be relayed onward and downward towards each other, called signal convergence. G protein convergence signals A, B, or any kind of two or three types of receptor will targeting single type of protein, but finally result is same, which is going to control the signal relayed onward to particular system called signal uh, convergence. Okay, this is the today's class on signal convergence and signal divergence. So signals may be vary, their origin is different, their receptors are different, but finally after secondary messengers they will leads to control two types of activities. What are they? Either cellular protein targets or nuclear protein targets. So cellular activity is controlled, gene expression activity was controlled. That's why it is called signal divergence, signal convergence. If any type of signals will share a common signal factors, that is called signal crosstalk. One signal pathway sharing the other signal molecules by two origins. That's why it is called signal convergence, signal crosstalk. Okay. Signal crosstalk plays a major role. In absence of particular signal only, next signal, next receptor also profoundly modulating the cellular activity, gene regulation activity. Thank you one and all for your patient listen.